Hi, welcome back to my channel. In this video, I will be talking about gluconeogenesis. So, gluconeogenesis is basically the production of glucose through any substrate which is not a carbohydrate. Alright, so let's just understand the pathway and then all of these questions will be answered. So, first of all, you need to know that first of all, so what happens is we have pyruvate. So this is mitochondria and this is cytosol. Alright, now what happens is in mitochondria we have pyruvate. Now pyruvate cannot exit mitochondria as such, so we convert the pyruvate into oxaloacetate by an enzyme which is pyruvate carboxylase. Then we convert the oxaloacetate into malate. Now this malate is able to cross and go into the mitochondria. So here we have malate and then we have oxaloacetate. So this is through an enzyme called as malate dehydrogenase. Okay, similar here. Then what happens is this oxaloacetate then further undergoes reaction and converts into phosphoenol pyruvate. Now the enzyme is important. Now this enzyme that you should know is phosphoenol pyruvate carboxykinase. Now all of the other reaction further occurs in the cytosol. Now I'll just enlist the steps that occur in cytosol. So basically we have to reverse the glycolysis. Okay, so see, pyruvate is now converted into phosphoenol pyruvate. Then this is converted into 2-phosphoglycerate. Then 2-phosphoglycerate is converted into 3-phosphoglycerate. Then we have 1,3-biphosphoglycerate. Then we had the two intermediates, dihydroxyacetone phosphate plus the glyceraldehyde 3-phosphate. Then after this we have fructose 1,6-bisphosphate, and then we have fructose 6-phosphate, then we have glucose 6-phosphate, and then we have glucose, the final product. Now you need to remember the names of the enzymes. So this is basically 3-phospho and 2-phospho. So if you see this is just mutase, right? So this is phosphoglycerate mutase, and then 3-phospho, this is this was phosphoglycerate kinase. Now we are losing one phosphate, so this was phosphoglycerate kinase. Then this reaction, the 1,3-BPG into this was glyceraldehyde 3-phosphate dehydrogenase. Now this is the reaction you should by heart the enzymes. Okay, then here we knew that the enzyme is our isomerase aldolase, and this was isomerase enzyme, right? Remember, now this reaction and this reaction, they are the important steps because they are irreversible. Okay, and the enzymes are different from glycolysis. So fructose 1,6-phosphate into fructose 6-phosphate is through the action of fructose 1,6-phosphatase, and this is through glucose 6-phosphatase. Okay, now the site is liver, kidney. Cell organelle is somewhat mitochondria and cytosol. Most of it is in cytosol. Major substrate, all the non-carbohydrate substrates like lactate, we have pyruvate, right, and so on. Pathway we have discussed. Now how the questions are asked about this. So first of all, the commonest question that they ask is why gluconeogenesis does not occur in in muscle in wait I think why gluconeogenesis does not occur in okay, leave this question. So first of all, the commonest question first of all they will ask is that the enzyme which is a biotin dependent activator is pyruvate carboxylase, and it is activated by acetyl CoA. Okay, then they will ask you the 3 or the 4 key enzymes of gluconeogenesis. So you will be asked, so first of all you should remember first one, all the different enzymes. Okay, so pyruvate, which were not in glycolysis, pyruvate carboxylase, then we had phosphoenol pyruvate carboxylase, then I told you about 2 enzymes which was fructose 1,6 bisphosphatase, and 4th was glucose 6 phosphatase. Okay, then all of this is done. Fine. So this is the pathway of gluconeogenesis, and yeah, they ask you why in liver and kidney. Yeah, the question that I was going to ask is why in liver and kidney gluconeogenesis is possible. So it is just possible because of the presence of the enzyme glucose 6 phosphatase. Okay, so I hope this video was helpful for you, and if it was, please subscribe to my channel, give it a like, comment, share, and thanks for watching.